Hi. So, in previous session we have learnt about the importance of innate immunity, and we have learned the various component of innate immunity. If you remember, there is a mechanical, physical or biochemical and microbiological barriers. Okay. So, we have discussed in a great length all these sections, that is mechanical barrier, the biochemical barrier and microbiological barrier. So, one more important, the most important component of innate immunity was left, that is cellular component. Okay. So, cellular component of innate immunity is basically consist of all cells. It is very interesting. All cells of the host plays a very important role in innate immunity, in addition to the professional innate immune cells. Okay. So, the cellular component of immunity is all cells plus the macrophages, the neutrophils, basophil and so on and so forth. Okay. So, now today I will introduce with the concept of this pattern recognition receptor, and this pattern recognition receptor is now playing a very important role in not only innate immunity, it is also playing very important role in development of whole adaptive immunity. Okay. So, this story was began in 1989 when one professor, his name is Charles A. Janvey. Okay. So, Professor Janvey gave this concept that the immune system, the innate immune system is not non-specific. Okay. So, this was before 1989. So, before 1989 people thought that this innate immunity is a completely non-specific thing, and this basically consists of macrophages and the cells. These macrophage cell or phagocytic cells take up the antigen and present this antigen to the T cell in order to develop a complete B and T cell dependent immunity. Okay. But in 1989 this concept was emerged and this concept was given by Janvey, and he said that, or he suggested that the immunity is not non-specific, they do have some broad kind of specificity, and this broad kind of specificity play a very important role in sensing the microbial pathogen, and then this is also playing very important role in activation of this innate immunity in order to produce innate immune cytokines, and that overall plays a very important role in development of adaptive immunity, okay, which is mainly dependent on B cells and T cells. So, this concept was basically given in one of very reputed symposia, which we call it as a Cold Spring Harbor symposia in quantitative biology. Okay. So, you probably may aware that this Cold Spring Harbor laboratories are located near New York. Okay. So, over this concept was given, and the symposia is very reputed symposia. Over there, this discussion about the DNA structure, codon and all those things were taken place, and there was a good debate among the scientists in order to take it forward. Okay. So, over there this concept was given, and this is given by Janvey, and the note of his all discussion is given in Approaching the Asymptote. This is the title, Evolution and Revolution in Immunology. Okay. So, this after emerging this concept, Charles Janvey and his group also worked toward this direction, and in 1996 before his work, the work of Julie Hoffman, who worked on Drosophila, okay, and he gave this his findings, which is published in Cell Journal, and he very elegantly showed that there is a some group of protein which plays a very important role in ventral patterning in Drosophila embryo, plays a very important role in defense against microbes. They are basically antibacterial, they are antifungal, okay. And all this work was reported in this article. As you can see, this was published in Cell in 1996, okay. So basically he suggested that there is a protein known as Spatzel, and there is another protein Toll, okay. These two proteins play a quite important role in development of antimicrobial immune responses in Drosophila, okay. Please note that previously these two proteins were reported to play important role in dorsoventral patterning in Drosophila embryo, okay. So this upon infection of bacteria and fungi, this protein known as Spatzel, this was cleaved by cascade of proteolytic activity, and this cleaved protein basically bind with a toll protein, okay, and then there will be a cascade of signaling, and that result to the activation of transcription factor diff or dorsal, okay, and that result to the expression of antimicrobial peptide known as dorsomycin. He also suggested there are two pathways, one is this toll pathway and another is IMD pathway. So IMD pathway uses another transcription factor known as relish, and that result to the expression of another set of antimicrobial peptides. As here you can see that there is a protein known as atacin, sapropin, defensin, dipterysin and dorsosine, okay. So all those protein eventually develop a antifungal responses, okay. So after this work, this basically established the observation of the Janeway's hypothesis, okay, and he in his group also they were working towards this, and his group identified the homolog of toll protein in human genome, okay, and they have characterized this protein in with some very simple and initial experiment. Basically they first fish out that gene by most probably by bioinformatic approaches, okay. After that these genes were cloned, and then they were characterized, and they found out that activation of this protein indeed induced the various innate immune cytokines such as TNF, IL-6, so and so, okay. So this work was published in Nature in 1997, and after that one more work was published, and this work was given by another group, and the group name is this is the group of Bruce Butler, okay. So they have found out that one strain of mice is resistant to the LPS, or it is hyporesponsive to the lethal dose of LPS, okay. And subsequent characterization result to the discovery of one very important mutation in TLR4, okay. At that time this name was already in place. This is toll-like receptor which is present in mammals, okay. So this this was further characterized. These mouse gene were characterized, and they found out that in the cytoplasmic domain of TLR4 there is a one mutation which changes the amino acid from proline to histidine, and that makes it resistant to the LPS lethal dose. Okay. Ideally, LPS, if you inject the LPS in the mouse or human, that will cause a shock. Okay, that's a very fatal condition. Okay, so they identified this mutation, and they have characterized this mice, and they found out that at 712 position there is a change in amino acid, and that makes the hyporesponsive to the LPS, and this gene is TLR4. So with this, it is quite well established that TLR are playing a very important role in sensing microbial pathogen, or at that time one more term was coined, that is a pathogen associated molecular pattern. Okay, so this is a pathogen associated molecular pattern, the LPS, or there are so many molecules I will discuss in subsequent slides. Okay, so after this this work received a lot of attention around the world, and then this work also received the Nobel Prize. And if you see, 2011 Nobel Prize was given for the discovery of this pattern recognition receptor to two persons. One is Julie Hoffman, who worked in Drosophila, and another person is the Bruce Butler, who characterized this TLR4. Okay, the hyporesponsive of TLR4 in particular strain of mice. Okay, and in addition, the Nobel Prize was also given to the Ralph Steinman. I have discussed earlier in previous session. He just passed away 3 days before the announcement. So this prize was given to his wife. Okay. So at this time, this concept was very well established, that innate immunity is not non-specific. The innate immunity do have a specificity, and this specificity is quite broad, and this broad specificity is basically playing important role in sensing and then subsequent signaling, and then development of innate immune response, and which eventually result to the development of adaptive immune response. Okay. So after this work, there are so many things to complete and finish in TLR field, and the most important contribution was by Professor Shizuo Akira. He is from Osaka University, Japan. He characterized various TLRs and TLR signaling molecules by creating by creating knockout mice, or by doing the genetic study. Okay, and this field was fully established at that time. So now I would like to talk about the innate immune response in Drosophila, because this is very important. If you see very carefully, the original discovery of phagocytes or phagocytic cells or phagocytosis was given by Emil Metchnikoff, which was discovered in a starfish, right? And after that, this discovery was made in various mammals, including human. Okay, so similarly, this work, Drosophila work, it's a great turning point in innate immunobiology. Okay, and this work result to the discovery of a whole new field of innate immunobiology, or there is a pattern recognition receptor and all those things. So I will quickly tell about the innate immune response in Drosophila. And if you remember, in early session I gave you the importance about the comparative immunology study. Okay, so this is one very good example. Okay, so let's begin with innate immune response in Drosophila. So basically these these Drosophila or fruit fly, they have a various cell for the defense which is phagocytic in nature, and these cells are basically fat body cells and hemocytes, and they take up the microbial pathogen and develop a appropriate immune response. And the appropriate immune response is basically mediated by antimicrobial peptide, okay, and these are as I showed you in previous slide in the work of Julie Hoffman, these are basically dorsomycin, which is a diff dependent. So what is diff? So diff, if you know that this diff is basically a transcription factor which is similar to the NF kappa B, okay. So NF kappa B plays a very important role in immunity, okay. So diff is just like that, and its other name is dorsal, okay. So diff dependent, there is a synthesis of dorsomycin, there is a another set of molecule which is synthesized under the control of transcription factor relish, okay. This is dipterysin, atacin and cypropin, okay. So and these antimicrobial peptides are generated through this IMD pathway. IMD is a immunodeficiency signaling pathway which is operated in Drosophila, okay. So but how these entities are sensed, that is bacteria which is consist of gram positive, gram negative, and there is a also fungi, okay. So what are the sensors? So the next question is that. So the sensors are there are a variety of sensors. The two major classes of sensors are peptidoglycan recognition protein, okay, and it is consist of 13 member, and basically it binds with a peptidoglycan of bacterial cell wall. There is another group of sensors which is we call it as a gram negative binding protein, GNBPs, and basically they recognize as LPS, which is a signature molecule of gram negative bacteria, and they also recognizes a fungi originated molecule, that is beta 1,3 linked glucans, okay. So glucan is basically present in fungi, okay. So in that they recognizes these GNBPs are basically play an important role in recognition of gram negative bacteria and fungi. So there are some set of molecule like GNBP1 and PGRPSA molecule. So they basically sense the peptidoglycan from gram positive bacteria, okay, and it recognizes and induces the synthesis of antimicrobial peptides. GNBP3 basically play a very important role in sensing fungi, okay. They recognize as fungi, and then that synthesizes the antimicrobial peptide. So how this is basically taking place, all this sensing and signaling. So here I will give you a quick glimpse. So basically what is happening, there is a activation of a serine protease, which is one serine protease known as GRAS is involved, and that triggered the proteolytic cascade that eventually end up with the cleavage of the inactive form of Spatzel protein, okay. And this Spatzel protein which is generally present in the cell, not cell, in extracellular space in an inactive form. So this is get cleaved, and then this active Spatzel interact with toll, and this basically makes a dimer, and this Spatzel make a dimer, and then this interact with or induce the dimerization of toll protein and induces the synthesis of antimicrobial peptide. Here you can see it's very nicely shown in this slide. In here you can see that there is a peptidoglycan, and this peptidoglycan is sensed by GNBP1 and PGRPSA, and that will, upon sensing, that will activate the protease cascade, and eventually this activate GRAS, okay, and this GRAS basically cleave the Spatzel protein and make a dimer, and that will make the dimerization of toll protein, and then eventually this will activate the downstream signaling, and towards end it will activate the diff protein, okay, which is closely associated with a protein known as a cactus, okay. This cactus is like a I kappa B in NF kappa B signaling, if you have studied. So NF kappa B is a transcription factor which is generally present inside the cell in bound form with I kappa B, okay. And once this I kappa B get phosphorylated, then this I kappa B will undergo proteasome mediated degradation, okay, upon after ubiquitination, okay, and then NF kappa, free form of NF kappa B will be translocated into the nucleus, where this will trigger the transcription of various relevant genes, okay. So this is a quite similar as human or mice, although the molecules are different, but they are quite similar, okay. So if you see very carefully, the toll is a molecule which is very important or pivotal in the defense of in defense against these microbial pathogen. However, they are not direct sensor, they are not pattern recognition receptor. This is very important note. Please remember, the toll is playing an important role in defense in case of all kinds of microbial pathogen in Drosophila, but it is not the sensor. Sensors are like as you have seen, there are several molecules or family of molecules that they play important role in sensing. Toll are basically control. Originally it was discovered as a controller for dorsoventral patterning in Drosophila embryos, and basically this toll get activated when Spatzel protein binds with the toll, okay. So what is Spatzel? So this is a little interesting. This Spatzel name was given due to the appearance of this molecule, and this is a one of German food, okay, which is made from the wheat. It's something like a spaghetti or macaroni or so on and so forth, okay. Towards end, I would like to just quickly give the glimpse of the toll pathway in Drosophila. Here you can see this Spatzel dimers are bound with toll protein, which eventually activate the diff or dorsal, and there is a cactus which is like I kappa B, and that activates the synthesis of antimicrobial peptides. This is a IMD pathway, where if you see towards end, there is a activation of relish, okay, and this basically start the synthesis of antimicrobial peptide. So in this session I will just stop here, and in next session I will talk about the talk in more detail about the TLR ligand and TLR signaling pathway and so on. So thank you.